So in continuation to the general characters of fungi or previous classes, so this is a specialized somatic structures and aggregations of hyphae. So what are the different kinds of specialized structures are there, what is the function and why the specialized somatic structures are formed from these hyphal aggregations and all those things now we'll see. So my name is N.H. Shankar Reddy, PhD plant pathology. So when it comes to thallus, that is a hyphae, so this hyphae is actually modified into various special structures in order to get the functional requirements done. For example, however, we have a different organs to get the functions done. So this hyphae actually modified or specialized into different structures to get the to done the different functions. So these are all the list of specialized structures: rhizoids, Appressorium, Haustoria and Hyphal traps. So now we will see in detail about one by one. So the first one is rhizoids. So rhizoids like hair-like structure or root-like structure that we can only observe this kind of the structures in Rhizophus. Only Rhizophus this kind of structures can be seen. So rhizoids are generally a short root-like structures or hair-like structures that we can observe at the Rhizophus. So rhizoids can be only produced by Rhizophus and Mucor, but no other fungi. So Rhizophus only can produce this kind of structures. We can also call it as anchoring organ or holding organ of Rhizophus. So this Rhizophus has functions as anchoring and also absorption organ. So it actually mainly gives anchoring or stability to the fungi so that it can hold, and it can also helps in the absorption of nutrients or absorption of various kinds of things that can be required for the fungus. So this kind of rhizoids or root-like structures can only observed in Rhizophus. The next one is Appressorium. The plural is Appressoria. So when it comes to the process of pathogenesis, now we will see in detail about how the spore will come and land on the leaf, and how it will produce germination tube, what is the function of Appressoria, how it will starts to like penetrate inside, and all those things that we will see in detail when it comes to pathogenesis. So we have a separate lecture on pathogenesis, no need to worry about it. So when it comes to Appressoria, generally Appressorium is like attachment organ of fungi. So why it need to produce another structure? So spore is already there, right? Why it is starting to germinate and producing a swollen globose like structure, or like lobed like structure, swollen lobed like structure. So the main function of Appressoria is to attach. It very strongly attaches the leaf surface, or it very strongly holds the leaf surface. So this can provide a stability so that from Appressoria the penetration peg will be developed. We can see here, this is Appressorium attachment organ. From the Appressoria penetration peg is going inside. So let's talk about Appressorium. Is generally lobed like structure or from the germinating hyphae. So the main function is adhere or attach the host surface. It provides a very strong anchoring or stability for penetration. So it actually, the penetration peg, as I told you, that is penetration peg is actually originated from this Appressoria. It's not only gives stability but also penetration peg, sorry, hyphae, penetration hyphae is actually developed from this penetration hyphae, that is a penetration peg is actually developed from Appressorium. For example, if you see Appressorium is produced by many kinds of pathogens like rust, powdery mildews, we can simply say it as an attachment organ, nothing else. So the next one is the Haustorium or Haustoria. So Haustorium is generally nutrient absorbing organs. This is spore, right? Is germinating and produce a bulb or swollen like structure called Appressoria. From the Appressoria, from that Appressoria, a penetration peg or penetration hyphae going inside. From penetration hyphae, a swollen, what can I say, this a swollen like structure or knob like structure which is penetrating inside the epidermal cells. That the structure that is penetrating inside the epidermal cells is known as Haustoria. So it is Haustoria is simply a nutrient absorbing organ or food absorbing organ or energy absorbing organ of fungi. So however we want balanced diet like proteins or energy, whatever it is, simply I would say energy, right? So however we need a sufficient amount of energy for our survival, fungi is also required continuous supply of nutrients, right? So that the nutrient exchange or the nutrient absorption can be done by a specialized structure called Haustorium or Haustoria. So this can be mainly produced by obligate parasite like Iridinales, Erysipelas and Peronospores. Other pathogens like Phytophthora also produce Haustoria and Appressoria, that is secondary. So actually the shape of the Haustorium may varies. So we will see in the next slide like knob shaped or elongated shaped or finger like various kinds of Haustorium can be observed when it comes to pathogens. So generally Haustoria means attachment organ. When it comes to Appressorium means attachment organ. Haustoria means nutrient absorbing organ and energy absorbing organ. So these are all the different shapes of Haustoria that is produced by the pathogens. Now I will tell you what is the shape on which pathogen can produce this kind of structures. So knob-like Haustoria is produced by albugo. So finger-like Haustoria is produced by Phytophthora. Branched or peg-like Haustoria is produced by Sclerospora. Bulb-like Haustoria is produced by Cuscuta. Wedge-shaped Haustoria is produced by Archithobium. Finger-shaped downy mildew, globose powdery mildew, pyriform Basidiospora. So these are all like different shapes of Haustoria. As I said, Haustoria is of different shapes. So these are all the different shapes of Haustoria. I think this slide was already explained in the previous presentations, I think. So the next one is Hyphal traps. So when it comes to Hyphal traps, this was especially produced by very few fungi like Dactylaria candida, Arthrobotrys. This kind of fungi can produce this Hyphal traps. The name itself indicates trap. So it can create a trap to the nematodes. We can see here it is creating a trap, especially Dactylaria. We can also call it as predaceous fungi. So predaceous fungi. So which means because it traps various kinds of nematode by using the aggregations or trap-like structure. If you see here, a thread-like structure or something or it strongly holds the nematode. So these modifications are generally, or these thread-like modifications are generally known as snares. Snares are Hyphal traps. So this kind of structures is actually produced by the fungi like Dactylaria candida, Arthrobotrys and Monocosporium. So this kind of fungi can produce Hyphal traps, otherwise called as predaceous fungi. So Hyphal modifications in fungi are mycelial aggregations. So far we have seen specialized somatic structures for various functions. Now we will see why Hyphae is modified into different kinds of structures and what are the different aggregations of fungi. So in order to get the functional requirement, fungi are, that is a Hyphae undergoes various kinds of modifications or certain kinds of modifications. So these are all generally, these fungal structures are maybe resting bodies, that is the resting structures or resting spores, maybe fruiting bodies or migratory strands. All these Hyphae aggregate into various kind of structures. Now we will see what are all they. So before that we need to understand a few things. So like what is plectenchyma, what is prosenchyma, all those things that we don't understand. So actually mycelium is generally organized into compact or woven tissues. How can I say that? Like loosely compact or woven tissues, that is generally called as plectenchyma. So this plectenchyma, modified into different kinds of fungal tissues that I will tell you what are they. So when it comes to this plectenchyma, basically is of two types. So first one is the prosenchyma, the second one is pseudoparenchyma. So on the right side I have given pictures prosenchyma. So here this is the Hyphae, right? This individual Hyphae are lies parallel to each other. For example, imagine, so this is a Hyphae. This Hyphae, this kind of many Hyphae lies parallel to each other, or like which individual lies are Hyphae lies parallel to each other, are interconnected to each other. This interconnection or loosely interwoven tissues, that is the Hyphae lies to each other, is called as prosenchyma. If his Hyphae is lying side by side, it is called prosenchyma. When it comes to pseudoparenchyma, on the right side we can see pseudoparenchyma. This is pseudoparenchyma, right? So it is actually loosely, we can see loosely compact or more or less isodiametric oval cells. So it is a compact, more or less isodiametric egg-like cells, like oval cells or egg-like cells. So this kind of structures can also observed in when you see the cross section of a plant or higher plants. We can see the same kind of structures, that is pseudoparenchyma. This plectenchyma is of two types, prosenchyma and pseudoparenchyma. Here this prosenchyma and pseudoparenchyma puts together forms various kind of structures. So this all various kinds of structures are actually made from this prosenchyma and pseudoparenchyma. Now we will see what are all the different kinds of Hyphal modifications are there. So the first one is the somatic or reproductive structures, that is first one is stromata or stroma. We can also call it stromata or stroma. It is a compact mass of tissues. If you see here, we can observe stroma. Like it is a cushion shaped or cushion shaped structure which is actually composed of pseudoparenchymatous tissues. So it is composed of pseudoparenchymatous tissues. So it is actually a fructifications or mattress like structure where the spores will be produced. Imagine, so it is a flask shaped or cushion shaped structure. Imagine, so this is like a fructifications, like a like a imagine, like a, there is a saying in in Sanskrit called Akshaya Patra, which means, so in that like continuous production of things whatever we want, so continuously it will produce this Akshaya Patra. So I can also say this stromata is like Akshaya Patra where continuous spore production will takes place, or fructifications of like somatic structures where continuous spore production or continuous production of this reproductive I mean spores will takes place here. So when it comes to say next one వచ్చేసి next one is sclerosium. So plural is sclerotia. So we can see here sclerosium, right? So sclerosium is generally a very hard, compact, resting structure that is actually happens due to aggregation of hyphae. I would say during unfavorable time, during unfavorable conditions, few pathogens, that I will tell you what are the few pathogens at the end. So during unfavorable time, few pathogens can aggregate this all hyphae, right? All hyphae can be aggregated or compacted like a very, like a globo structure, very hard, compact mass of structure can be formed by this aggregation of hyphae. So this is also actually composed of pseudoparenchymatous tissues only. It is a hard, compact, resting structure that is produced by the few pathogens during unfavorable environmental conditions. So this can survive during unfavorable time. Whenever the favorable time occurs or favorable environmental conditions occurs or onset of favorable environmental conditions, the same sclerotia. What is sclerotia? It is just aggregation of hyphae. This is a compact aggregation or globos, or like a compact globos or aggregation of just hyphae, right? So whenever the favorable time happens, what will happen is automatically hyphae starts to grow and will produce another mycelium or maybe like fungi, right? So this sclerosium can survive in soil. Mainly sclerosium is actually survives in soil. So during unfavorable time, how it will survive, I will tell you. So it contains a huge amount of reserve food material. I will tell you one more example. For example, if humans can't eat for 2 to 3 days, we have a separate organ called adipose tissue where energy can be stored in the form of fat. So whenever we are not getting food or whenever we are not eating, the energy can be utilized from this adipose tissue. So here during unfavorable time, it won't get the food from outside, right? So that it contains a huge amount of food materials inside that was actually packed in a complex form like a mannitol, trehalose, especially glycogen and lipids. So when it comes to fungi, the food storage organ is in the form of glycogen and lipids, so which is actually helpful for a long time survival during unfavorable environmental conditions or harsh environmental conditions. Whenever the onset of favorable environmental conditions, again the sclerosium starts to germinate and can produce fungi. So the pathogens like sclerosium or sclerotinia, sclerosium rholpsi, claviceps, rhizoctonia, macrophomina, these pathogens can produce a resting structure called sclerosium during unfavorable time. So anyway, let me come once again in a short form. This is a hard, compact, resting structure that is aggregation of hyphae which is produced by the few pathogens. So the few pathogens are Sclerotium ralsi, Sclerotinia, Claviceps, Rhizoctonia, Macrophomina. These are all the few pathogens which produce sclerosium as a resting structure. So this contains, the sclerosium contains high amount of reserve food materials in the form of glycogen or lipids in the main forms. So this can be helpful for long time survival during unfavorable times or harsh environmental conditions. Whenever the onset of favorable environmental conditions, again this Sclerotia starts to germinate and gives rise to mycelium or new fungi. So actually, I would like to tell you one more, important point about the sclerosium. So the sclerosium of Australian polyporous millete, that is now it is called Lacocephalum millete, can reach a size of human head. Normally, the sclerosium can't grow more than a size of mustard. is exactly look like a mustard. If you see the cultures, if you see the culture here, we can see exactly look like a mustard, right? Exactly look like a mustard. So this won't grow more than a size of mustard. If you see some sclerotia like a polyporous militae, that is Rhizocephala militae, so it can reach a size of human head size, about 15 kilometers, so sorry 15 kilograms. So it is also known as a native bread or blackfellow's bread. So as I said, like sclerotia can survive during unfavorable time in the form of glycogen or proteins or lipids that can be stored inside, so that it can be helpful for long term survival, right? So this is generally about sclerotia. So when it comes to the next one, mycelial strands, the name it indicates, all strands of mycelium are put together is called mycelial strands. So the strands actually happens parallel to each other, lies parallel to each other, side by side, as like of prosenchyma. But here formation or of aggregation of a parallel relatively undifferentiated hyphae. If you see here, you can't differentiate, it's a completely undifferentiated, we can't find where exactly the mycelial strand came from. So this kind of structures can be seen commonly in Basidiomycota, Ascomycota and Deuteromycota, that is a higher fungi. Okay, so this kind of structures can have especially observed in higher kind of higher fungi. So in one more important thing is when it comes to this mycelium or this parallel aggregation of hyphae, where nutrients can be exchanged or passed or translocated in a both direction, upward direction as well as both upward and downward direction. If you see in general, if you see in in I think in third class we discussed about general characters of fungi. So this is also in continuation to general characters of fungi only. So there I told you in Hyphae, I know there is Spitzenkarper will be there, where continuous synthesis of vacuoles or energy materials will be takes place, so that Hypha goes in upward direction. When it comes to mycelial strands, energy will be flowed in both direction. I repeat, in mycelium, general mycelium or Hyphae, energy will be flowed in only one direction, that is upward direction, so that hypha starts to germinate. If hypha starts to germinate, automatically the mycelium also increases, right? So when it comes to this mycelial strands, energy can be translocated into both the directions. That is one important thing. So for example, circular lacrimans can such kind of fungi can be can produce this kind of structures. So one important point, as I told you, that this mycelial strands is capable of translocating the nutrients and water in both direction. So when it comes to the next one, rhizomorphs, otherwise called as mycelial cards. It is simply aggregation of somatic hyphae. So here all the somatic hyphae aggregated into this specialized structure called rhizomorph. Here this rhizomorph contains many aggregations as of an average of 1000 hyphae. So 1000 hyphae, an average 1000 hyphae aggregated and form a specialized structure called rhizomorph. So in some dead trees we can observe up to 4 mm in diameter of rhizomorphs can be observed. So it generally contains 1000 aggregations of hyphae. 1000 hyphae can be aggregated and form a structure like this. So this kind of structures can be observed when it comes to Basidiomycota as well as Ascomycota also. Best example is Armillariella melia. So it is called as honey fungus or bioluminescence fungus or honey agaric. So I will discuss in detail about these mushrooms and all those things, because we also have a planning to separate classes about only mushrooms. So various kinds of mushrooms, beneficial mushrooms, non-beneficial mushrooms, all those things, poisonous mushrooms and all those things, we have a separate division. There I will discuss in detail about each and every single mushrooms. So when it comes to in general, rhizomorphs is produced by the mushrooms like Armillariella melia and Ganoderma lucidum. So this is also a resting structure. Whatever we have seen, this special Eiffel modifications, all are resting structures. So mostly resting structure, like sclerotium is a resting structure, rhizomorph it is also a resting structure. Okay, and one more thing is also there, that chlamydospore, however, like a sclerotium, chlamydospore is also a hard resting structure that is produced during unfavorable time. So sclerotium is generally produced by the pathogens like Sclerotium ralsei, Macrophomina. So these are all the pathogens. When it comes to chlamydospore, that is produced by the pathogens like Fusarium. Okay. So, and one more important point I would like to tell is an Armillariella hostia, a specimen from a Melhur National Forest in Oregon. It covers 3.4 square kilometers. Only one Armillariella area, I am telling. one mushroom covers just 3.54 square miles. So this hyphal modifications or network connects one root to another root, and in ground level it will goes on and goes upon go on and goes on. So 3.4 miles can be covered with this only mushrooms. And one more thing, Almillariella gallii, a specimen in Michigan, covering 37 acres. 37 acres it covers. So the remaining general characters will be continued in the next videos. So this is one of the book that was published in Kalyani Publishers. The book name is A Vision into Plant Pathology, a complete student version. So when it comes to students, I believe it is a student encyclopedia. I hope this book will be very useful. If you want extra information or extra knowledge, or if you want to pursue your career in plant pathology, I suggest you to go with this book. For further information, students can reach at www.geekyresearcher.com. Stay geeky and stay tuned. We are Team Geeky Researchers.